My name is Shara Babcock. I serve Chautauqua as Vice President and Emily and Richard Smucker Chair for Education. It's my great pleasure to welcome you to this week's Chautauqua Literary and Scientific Circle Roundtable discussion with Ed Yong, author of I Contain Multitudes, The Microbes Within Us, and A Grander View of Life. This book is a part of our season-long book theme of community, explaining the community of microbes that lives with each of us. A couple of announcements first. Out of respect for Ed Yong and for people in the audience with you, please silence your cell phones and all other electronic devices. And as is our custom, Mr. Yong will be available for a book signing immediately following this presentation on the porch of the Literary Arts Center at Alumni Hall, located across the grove from this venue, right straight through there. This concludes the afternoon announcements. Today's presentation is made possible thanks to generous support from the LGBTQ and Friends community, whose mission is to expand diversity within the greater Chautauqua community for LGBTQ people and to educate others through dialogue about the LGBTQ community and their experience here and elsewhere. The Chautauqua Literary and Scientific Circle began in 1878 as an educational venture to provide learning for adults without access to college. It has become our nation's longest-running continuous book discussion group, and upwards of 100 people graduate from the program each year. Each class designs a banner, and I have chosen the banner to support today the CLSC class of 2002. Their slogan is aspiring towards greater comprehension. This banner features a real three-dimensional representation of DNA. And those of you on this side, after the lecture, you might want to come see this banner. It's usually in Alumni Hall. It's very hard to travel with because it is woven and it is very heavy. As a book that introduces us to the fascinating world of microbes, I Contain Multitudes does exactly that, aspiring towards greater comprehension. It takes readers towards a deeper awareness of our complicated yet beautiful personal ecosystem. Published last year, I Contain Multitudes presents a microbe's eye view of the world, revealing a fascinating and original understanding of life on Earth. Since every animal is home to millions of bacteria and other microbes, Ed Yong prompts us to look at ourselves and our animal counterparts in a new world and in a new light, less as individuals and more as the interconnected, interdependent multitudes we undoubtedly are. Bill Gates deemed I Contain Multitudes to be science journalism at its finest. Ed Yong is an award-winning British science journalist. He is a staff writer at The Atlantic and perhaps most well known for his former blog, Not Exactly Rocket Science, which was published as part of National Geographic's blog network. For his work, Mr. Yong has received honors, including the National Academy's Communication Award and the National Union of Journalists Stephen White Award. I Contain Multitudes is Mr. Yong's first book. So, ladies and gentlemen, scientists, physicians, and humanities scholars, diversity experts, and the microbes around us, please join me in welcoming Ed Yong to Chautauqua.
Hi, everyone. Okay, so I'm going to start this talk by taking you back in time by about 350 years, to the year 1675, and across the ocean to the town of Delft in the Netherlands, where a man named Anthony van Leeuwenhoek is about to change the world. Van Leeuwenhoek is not a scientist, he is not a scholar of any description, he is just a civic servant and a humble draper. But he has two qualities that are very important for our story. He is exceptionally curious about the world around him, and he is also, for his time, the maker of the world's most powerful microscopes. He makes them himself. He grinds impossibly perfect spherical lenses, which he sandwiches between two brass plates to create an object that's probably slightly smaller than my cell phone, and that looks really quite unlike a microscope. It looks more like a glorified door hinge. But this innocuous object is capable of magnifying things far better than anything else around at the time. And Leeuwenhoek turns it onto everything else around at the time, onto bits of animals and plants, onto bits of soil and water. And water in particular, he enjoys looking at. He grabs it from the canals and rivers of Delft, from pots and urns that have gathered rain outside his house. And everywhere he looks at drops of water under his microscopes, he sees the same thing: life. In extreme abundance, microscopic living things that no one had ever seen before and that no one even knew existed. And yet there they were, swimming around, moving, clearly animated, and in untold numbers. And they existed everywhere, including within his own mouth. Because scientist that he was at heart, Leeuwenhoek also scraped bits of dental plaque from between his teeth and examined that underneath his microscope. And again, he saw these living things. And they were bacteria, microbes. And Leeuwenhoek had just become the first person in the history of all of humanity to understand that these things existed. And that, to me, must have been one of the most glorious and important moments in the history of biology, because microbes are the vast majority of life around us. And for most of humanity's history, we had no idea that they were there at all, much less that they were in us and on us. Now, since Leeuwenhoek's time, microbes have earned this rather negative reputation. We have come to view them as agents of dirt and disease and decay. And that started because a few decades, centuries even after Leeuwenhoek's seminal discoveries, a lot of scientists very rapidly showed that many of the most infamous plagues to affect humanity, things like plague and typhoid and cholera, tuberculosis, leprosy, gonorrhea, syphilis, some of the worst diseases to afflict people were all the work of bacteria. And so for the longest time, we came to view these living things as germs, as villains, as antagonists that we needed to destroy lest they destroy us first. And that framing still continues to this day. You will see countless newspaper headlines in which people swab surfaces around them, a wallet, microphone, laptop, this banister here, and grow them up and find that bacteria are living upon those surfaces. And they are all, this exercise is almost always taken as a sign that such surfaces are dirty, that we have failed to adequately clean them. And that is a gross misconception. It is misleading and it is unfair. Because the vast majority of microbes do not, in fact, cause disease. Only a few hundred kinds are known to do so. The vast majority of them are either benign or indeed beneficial to us. And in fact, bacteria are the lords of the planet. They have been around for the longest time, and they are around in far greater numbers than any of us can ever hope to achieve. Their presence isn't a sign of dirt. Their presence is just a sign that something exists. If you find surfaces without bacteria, it's probably a sign that something truly horrible has happened. I say that they have been around for the longest time, and I'm going to move away from the mic now and just shout, so bear with me. If we consider the entire history of the planet, all 4.5 billion years of the Earth's history to be represented by the length of this white facade in front of me, so that the Earth was created at that corner, and that we are now here, well then humans evolved around here. And all the animals and plants and the organisms you're familiar with, they evolved around here. The dinosaurs ruled from about here to here. So for the vast majority of life on Earth, since it first originated roughly around here, it was entirely microscopic. You would not have been able to see it with your own eyes, which would be fine because eyes only evolved here. So this is a bacterial world. It is a world that has been ruled for microbes for the longest time, and we are latecomers on the scene. We are just icing on this grand bacterial cake. And so when we, and by we I mean the entire animal kingdom, when we arrived on the scene, we arrived in a world that was full of microbes, and we evolved to live with them, to exploit them, and to depend upon them. To this date, each one of us sitting here today contains roughly 39 trillion bacteria and other microbial cells within our bodies. That is roughly one for each of our own actual human cells. So we are all just half the people we think we are. So when you see me standing up here right now, I look like just one individual, but in fact I am an entire world. I am an ecosystem, a vast community of life. And in fact, I am a vast number of different ecosystems. The dry skin on my forearm is parched and dry like a desert, whereas my mouth is moist and humid like a rainforest. And each of these body parts contains vastly different microbial fauna, just as rainforests and deserts and coral reefs house very different species of animals and plants. And this principle extends throughout the entire animal kingdom. So if any of you use bathroom sponges, many of those used to be animals too. They are the remains of some of the simplest animals in the world. Sponges are just a couple of layers of cells sandwiching a bit of jelly. But they too contain thriving microbiomes, thriving communities of microbes. An ant colony can number in its millions, but every single ant within that colony is a colony in its own right. If you look at a nature documentary of a polar bear walking alone in the Arctic wilderness, that polar bear is surrounded and full of life. When bar-headed geese fly over the Himalayas, they carry microbes with them. When sperm whales dive to the deepest oceans, they carry microbes with them. And when Neil Armstrong and co set foot on the moon, they were also taking a giant step for microbe kind. But these bacteria, these microbes, are not just passengers. They're not just stowaways in our bodies. They also provide critical support and really important benefits to their hosts. And these vary from creature to creature. One of my favorites in the book is an animal called the Hawaiian bobtail squid. It's a little squid. If it was in this room, it would sit on the palm of my hand. This animal has two organs in its undersides that house glowing bacteria. And the light that they produce shining down below the squid perfectly matches the moonlight that shines down on top of it, so that any predator looking at this animal from below can't make out its silhouette. Because of its bacteria, it has a kind of invisibility cloak. There are countless other examples of extraordinary powers bestowed onto animals by their hosts. There are flatworms that can regenerate lost body parts if they contain the right bacteria. There are birds that paint their eggs with defensive pastes of bacteria that stop diseases from afflicting the chicks within. Hyenas also release pastes full of microbes, and those release odors and scents that reflect the animal's sex, social status, and age. These are animals that can write their autobiographies in bacterial odors. I've been writing about this area of science for over 10 years now. And it's one of the most fascinating bits of biology to me, because I think for several reasons. It touches on everything. Everything that we thought we knew is actually uprooted and changed by our understanding of the microbial world. And in many ways, microbes are the ultimate underdogs. They have been feared and neglected for the longest time, and only recently are we starting to appreciate how important they are for our lives, which makes really good fodder if you are a professional storyteller. And indeed, many of the most familiar aspects of our lives take on this very wondrous and unfamiliar new air if we consider it through this microbial lens. So as an example, and one of my favorite examples, involves breastfeeding. So breastfeeding is obviously a way for a mother to nourish her child, but it is also much more than that. Because breast milk, about 10% of breast milk consists of sugars, very complex sugars that babies cannot digest. Those sugars are there to nourish microbes inside the baby's gut, and particularly certain strains of microbes that have evolved to digest those sugars with incredible efficiency. And they, in return, nourish the baby's gut cells, they seal up the lining of the gut, and they reduce inflammation. And so when a mother is feeding an infant, she is also feeding these microbes, including a strain called B. infantis. She is not just nourishing one individual, but sculpting this entire world, engineering an entire ecosystem inside her child. And in doing so, she sets up her child for life, because those communities of bacteria, as I said, don't just go along for the ride, they also contribute to the sculpting and the development of their hosts. So all of us, and indeed every animal, develops from a single fertilized egg into this adult body comprising trillions of cells. And that process of development doesn't just unfold under our own steam, under the instructions encoded by our own genes. It in fact happens in conversation and in negotiation with all the microbes that share our bodies. And we can understand that when we see what happens when it doesn't happen. There are many scientists around the world who can raise animals in germ-free conditions, meaning that for their entire lives they are raised in sterile bubbles disconnected from the microbial world. They do not contain multitudes. They are just individuals and nothing more. They're like a silhouette that hasn't been filled in. And as a result, these animals have a multitude of different problems. Mice that are raised germ-free, for example, will have deficiencies in their blood vessels, their bones, their guts, their immune systems. They'll behave differently. And that's testament to how important microbes are for sculpting and shaping our organs as we develop, and for influencing the continual growth and renewal of our bodies. I mentioned the immune system there. That's an interesting thing to talk about, because the immune system is traditionally viewed as a safeguard against microbes, a barrier. The typical metaphor is that of a militaristic police force that hunts down and destroys anything that is foreign or other, and that includes bacteria and other microbes. But obviously, we have large communities of microbes in our bodies that aren't being regularly mown down by the immune system. And indeed, many scientists have shown that those microbes are important for stimulating the growth of different branches of the immune system. So it's not just that the immune system tolerates the microbes within us, but that it is also shaped by those microbes. So I think a better analogy is that of a group of park rangers in a national park whose job is to care for the denizens of the park, to maybe keep their population levels in check, but also to chuck out invasive species and to make sure that everything is ticking along nicely. And in a final interesting twist in this developmental story, there are many studies that have now shown that, at least in things like mice and rats and other laboratory animals, the microbes that they carry can influence their behavior, their brains, the way they think and act. act. And that is a very disconcerting notion to us because we're used to thinking that our behavior and our choices are the result of ourselves, our brains, our own free will. And yet from animal studies, we see that depending on which microbes a mouse or a rat contains, you see differences in its personality, in its mood, in its susceptibility to stress or anxiety. Whether those studies translate across to humans is still an open question, but I think it would be very surprising to learn that we also are not influenced by the microbes within us. As I said, they play such an important role in our lives and in our development and in every aspect of our biology. All of these things that I've been talking about matter over the course of an individual's lifespan. They influence our health and our lives as we grow up. But these partnerships between hosts and their microbes have also huge consequences on evolutionary timescales for the fates of entire species and for entire animal groups. I think the answer is probably most of you, but does anyone here do gardening? Oh, good. Sometimes I ask that question in places like New York, and people are like, what even is a plant? All right, so you're all avid gardeners. You will have almost certainly had to deal with things like aphids, sap-sucking bugs, leafhoppers, many nods in the audience. Right. Sap-sucking bugs are part of a group of insects that are extraordinarily successful. There are 85,000 species of them, and they all drink the sap of plants. And they can only do that because they have bacteria inside their cells that provide them with nutrients that are missing from that diet of sap, essential amino acids that they would not otherwise be able to get. If those insects had not formed partnerships with those microbes at a very early stage in their evolution, they would not exist now. They would not have been able to tap into this enormously bountiful source of food and have radiated into those 85,000 species that give you all such problems. There are many other examples of this extreme dependence, and one of my favorites exists in the deep sea. So imagine sailing thousands of miles off the coast and then hundreds of miles down into the depths of the ocean, into hydrothermal vents, these like hellish Hadean landscapes where churning, boiling water spews out of undersea volcanoes. These are worlds with no light and precious little food, but there are still animals there. And they survive in those inhospitable worlds because they have microbes in their bodies which provide them with 100% of the nutrition that they need. Microbes that yank chemicals from those volcanic environments and use them to make nutrients in the same way that the plants around you use sunlight to create nutrients from carbon dioxide and water. And because of their partnership with these microbes, many of these animals, including clams, mussels, giant tube worms, have lost their guts, their mouths, their entire digestive systems. They do not need to eat because they have bacteria to provide them with all of the sustenance that they could possibly need. There are many more familiar animals that, of course, have kept their digestive systems, so things like cows, goats, sheep, or livestock that we all depend upon. They have converted their guts into gigantic fermenters, into vast reactors in which microbes can break down food. And they need those microbes because we animals are ill-equipped genetically to digest many of the tough carbohydrates and fibers in the plants we eat. And so something like a cow gets 70% of its nourishment through the microbes that live in its body and that help it to digest its food. Again, if none of those microbes existed, if those animals had not formed partnerships with those bacteria, then they would not have existed. Then the farms and ranches that we have would be empty. The vast plains of Africa would be devoid of the herds that, you know, when you do a natural history talk, you get very easily distracted by funky insects crawling on your podium. Excuse me for a second. Those too would be empty. Microbes also contribute to the evolution of animals not just by giving them new opportunities, but by allowing them to adapt to new challenges with extraordinary speed. So there are insects that can become instantly resistant to insecticides if they swallow the right detoxifying microbes in the soil around them. There are rats and mice that can become instantly resistant to toxic plants and so gain access to commonly available sources of food, again, if they swallow the right microbes. There are some of those pesky aphids that cause you so much bother can become resistant to parasites if they trade microbes through sex, making that one of the few examples in the natural world of a desirable venereal infection. Humans also benefit from some of these exchanges. So one of my favorite stories involves the people of Japan. For centuries, people in Japan and some other parts of the world have harvested seaweed from the coasts. Today, that seaweed is roasted to make nori, the stuff that wraps your sushi rolls. But in centuries past, people would eat nori raw, and because of that, they would swallow microbes that happened to live on that seaweed and were very good at digesting that seaweed. Seaweed consists of certain carbohydrates that aren't found on land plants and that are difficult for even land bacteria to digest. But ocean bacteria have the right tools for doing so. And when those ocean microbes hitchhiked a ride into the guts of people who used to eat seaweed, they encountered gut microbes. And they did something that bacteria do all the time that is very part and parcel of their everyday life, but that is extraordinary for us. They exchanged their genes. So you and I can only pass down our genes vertically from parent to child. But two microbes can just sidle up to each other and exchange their DNA just as easily as you or I might exchange gifts or conversation or phone numbers. And that happens all the time, and it happened in the bodies of these ancient seaweed eaters. And that is why, to this date, the gut microbes of people from Japan and some other parts of the world contain genes that allow them to digest the carbohydrates in seaweed with unusual efficiency, because those are loans from marine microbes. Because these types of horizontal gene transfers allow bacteria to evolve at an incredible pace, and they allow the hosts of those microbes to also pick up new adaptations very, very quickly. So, having gone on for some length about how cool microbes are and how important microbes are, one thing I want to stress to you is that microbes are not necessarily our friends, nor are they necessarily our enemies. They're neither good nor bad. There is no, in fact, such thing as a good microbe or a bad microbe. These are terms that are reserved for marketing campaigns with little biological relevance. In fact, it's best to think of us as just another habitat for them, another world, another drop of water, another lump of soil in which they can live. And many of us, humans, other animals, have found ways of coexisting with the microbes within us and certainly forming harmonious relationships with them. But those relationships can also turn sour. And the partnerships we have with our microbes can shift from positive ones into negative ones. There is a long laundry list of diseases and disorders that have been linked to changes in the human microbiome. A list that includes such things as diabetes, heart disease, colorectal cancer, inflammatory bowel disease, asthma, allergies, obesity. It goes on. And in many of these cases, the idea is not that a microbe is causing a disease in the way that a bacterium causes tuberculosis or leprosy or cholera. The idea is that an entire community of microbes has shifted into a negative state so that their interactions with their host become dysfunctional. And one of the terms of this is dysbiosis, the idea that a community relationship has gone awry. But I want to stress, because there is a lot of hype around the human microbiome, some of it warranted and some of it not, that many of these connections are still unclear. We do not know if the changes in the microbiome are the cause of the disease, whether they are a consequence of the disease, whether it is both or neither. And there are, however, some types of experiments which can help to answer these questions. Some of them involve those germ-free animals that I told you about. If you take a mouse or a rat that has the rodent equivalent of some of these diseases, say a mouse version of diabetes or heart disease or some such, and you take their microbes and put them into a germ-free rodent, then that recipient will often develop many of the symptoms of the conditions that its donor had. A sign that the microbes within that animal were not just going along the ride, but were, to a degree, grabbing the wheel. And the question for scientists now is, to what extent does that matter? Are these changes in the microbiome initiating these conditions? Are they just exacerbating them? Are the changes that we're seeing in laboratory studies relevant to us in the wider world? And to what extent do changes in the microbiome explain the prevalence or the rise of many of these conditions which have become more common over the 21st century? And I am pushing these to you as questions because that is exactly what they are. They are things that are the subject of active investigation rather than things that I have hard, solid answers to tell you about. Also, the subject of active investigation is the question of what actually changes the human microbiome in the first place, and what sort of things might disrupt it. There are many possible answers to this. We live in a very different environment than we used to, in urban settings rather than rural countrysides full of soil and animals, where we had access to a much more diverse range of microbes than we previously did. We have waged a very successful war on the microbial world. We use antibiotics to kill microbes that cause diseases. And I want to stress that antibiotics have done a huge amount of good for us. They have brought a number of very important diseases to heal, but they are also unsubtle weapons that harm microbes that we depend upon, as well as those that we do not, and that might cause us harm. Small doses of antibiotics are probably fine for us. The microbiome, like any other environment, like a burnt forest, will bounce back from injuries. But perhaps long chronic doses might lead to more long-lasting problems. In addition to that, we sanitize everything. We use hand gels and soaps and cleansers that contain all kinds of antibacterial chemicals, often to a degree where they are simply not necessary. We also eat very different diets than we used to, especially those of us in Western societies, we eat much less fiber than we used to. And that's important because fiber, being plant matter, contains a wide variety of carbohydrates, which nourishes an equally wide variety of microbes in our guts. And by cutting back on fiber and eating a wider range of animal foods instead, we have caused this winnowing of the diversity of microbial life that share our bodies. What can we do about that, if anything? Theoretically, we can do a lot. One of the reasons people are attracted to the microbiome is the idea that it is mutable, that it can be changed, that unlike the genes that we have, which are fixed from birth, the microbiome is something that is dynamic and changing all the time. And perhaps it is a lever that we can more easily push upon in order to improve our health. At least that's how it works in theory. In practice, it turns out to be very, very complicated to change these worlds that live inside us. So one of the most common ways people think about doing so is by taking probiotics, which are products that supposedly contain beneficial microbes. They have a very long history, and they have a huge amount of marketing hype surrounding them. Unfortunately, probiotics are actually very medically underwhelming. According to good, rigorous studies, they are decent for things like infectious diarrhea and for the types of infections that happen after long doses of antibiotics. But they do very little for many of the conditions that I've talked to you about, many of the conditions that are linked to the microbiome. And they actually do very little to shape the composition and the behavior of the microbes that live within us. Why is that? Well, for a start, many of the species and strains of microbes inside probiotics were chosen for historical reasons, because they were easy to grow and to manufacture, and not, in fact, because they are common inside the human body or particularly well suited to life within us. Many of them have been grown in industrial cultures for a very long time, so they are effectively domesticated. They are found in these products in very, very small quantities, a minute fraction of that which already lives inside us. And for that reason, taking probiotics is really a bit like going to a zoo and saying, it's like going into a forest like this and saying, here are a handful of animals that I have reared in a zoo and raised in captivity, and I'm going to release them in the wild and hope that they take over and flourish. And it turns out that they don't, for really obvious reasons, because if you take things that are ill-suited to these wild environments, they don't flourish. And so when we swallow probiotics, they don't do very much, and they pass out of us very quickly. So perhaps a much better solution to the idea of microbial medicine is to give people large communities of microbes that are, in fact, well suited to life in the human body. And this is the idea behind a rather gross and unorthodox treatment called the fecal transplant, which is exactly what it sounds like. It is a way of treating illness by giving a recipient the stool of a donor. For those of you who are wondering how this works, it is very, very simple. You can do it with a blender and a funnel. It sounds kind of awful, but the principle behind it is like returfing a lawn that has been overrun with weeds, and replacing it with fresh green pasture. And in this metaphor, the weed is typically a bacterium called Clostridium difficile, or C. diff, a bug that causes very severe, recurring, sometimes fatal cases of diarrhea. Fecal transplants seem to be the single best way we have of treating C. diff infections. In a lot of clinical trials, antibiotics only successfully cure around a quarter of C. diff infections, whereas fecal transplants have typically cured upwards of 90% of them. So unorthodox though they may be, they do seem to work in this particular instance. And because of the success of using fecal transplants to treat C. diff, they've been tried for a large number of other conditions, things like inflammatory bowel disease and irritable bowel syndrome and diabetes and all of the other things that I listed. But in those cases, the success rates are much lower and far more inconsistent. And in cases where scientists have tried to remove the ick factor by taking group cocktails of microbes that are naturally found in our stool and growing them up and giving them to people in the form of pills and capsules, again, we see unclear signs of effectiveness. And I think that just goes to show that even when you're thinking of moving an entire community of microbes from one person to another wholesale, it is very difficult to reset someone's internal ecosystems. And we still don't entirely know exactly what it is about fecal transplants that makes them so efficient for C. diff, what makes them work sometimes in other conditions and not on other occasions. And it just goes to show that we are at the very earliest stages of understanding these worlds inside us, even though there's been 350 years since Leeuwenhoek first looked down his microscopes, and that we are still struggling to harness this world for our own benefit. When we think about medicine, we often treat it in terms of very simple arithmetic. If you have a vitamin deficiency, it's because you lack something, so you add in that thing and you are better. If you have a viral infection, you have a surplus of a thing that's making you sick, so you subtract it and then you're better. But the mathematics of microbiome medicine are very different. This is a case not of simple addition or subtraction, but of changing and tweaking an entire world, a world that can be as complicated as a forest or a lake or a coral reef. And it is no wonder that an act of ecosystem engineering of this kind should be very complicated. It should be difficult to get right. So in terms of using the microbiome for our own good, I think there are a couple of ways in which we might be able to score some quicker wins without the need to change the communities within us or getting probiotics to work or anything like that. One might be to use them to personalize our medicines. So many of the drugs that we take, from common things like acetaminophen to complicated things like cancer immunotherapies, work better in some people than others depending on the microbes that we carry. The bacteria might render the drugs harmless by metabolizing them. They might convert the drugs into something toxic that causes side effects. So I think in the near future, we might get to a point where looking at someone's microbiome will allow doctors to more to prescribe medicines in a more fine-tuned way. We might also be able to drug the microbiome, to use medicines that change the way the bacteria inside us behave without necessarily changing who is there. So there are, for example, medicines that can alter microbes that contribute to the risk of heart disease. Many of you might take statins. Anyone take statins? Okay, statins are drugs that reduce the risk of heart disease by lowering cholesterol. There are other drugs that might be able to do the same thing by attacking not human enzymes like statins do, but bacterial enzymes. So maybe in the future, doctors will prescribe you, if you have a high risk of heart disease, not just a statin for the human half of you, but another drug for the microbial half of you. But again, and really this is the most important part of this talk, other than the fact that bacteria and microbes are actually really cool. We have a long way to go. We barely understand, we barely have a full list of all the species that live within us. We cannot still account for why the microbiome varies so much from person to person. We know that it's influenced by things like what we eat, the medicines we take, the exposures we had as children, the way perhaps our genes, our immune systems, the contacts we have with other people. But if you put together all of what we have learned, we can only explain maybe about 10% of the variation in the microbiome across different people. Our microbe communities vary substantially. Mine is very different to yours and yours and yours and yours. And the vast majority of those differences are still inexplicable to us. And if we go into the broader world, our depth of ignorance becomes even greater. Our understanding of the microbes in other animals, in plants, in water, in soil is still very, very shallow. And I think it is important to understand more about that, which is why I Contain Multitudes is not just a book about the human microbiome. It is not a self-help book or a diet book. It is a book about natural history. It is a book about the world around us, the creatures we share it with, and the creatures that they share themselves with. And that matters because sometimes the exploration of the natural world for its own sake leads to incredible and unexpected benefits. It was the exploration of the natural world that led people like Charles Darwin and Alfred Russel Wallace to come up with the theory of evolution, the single most important biological idea of all time. And it is the exploration of the natural world that is the subject of the final story that I'm going to tell you today, which is about microbes and the unexpected benefits that they can provide to us. So it begins in 1924 when two scientists named Marshall Hertig and Simeon Wolbach discovered a new species of bacterium living in the cells of some insects that they had collected outside Boston. The two of them had no idea what this bacterium did, what it was, whether it was important, and so it took them 12 years to even bother giving the thing a name. And Hertig named it after his friend, Wolbach, which is why today that bacterium is called Wolbachia. Wolbachia turns out to be extraordinarily successful. It is found in something like 40% of species of insects and other arthropods, which are themselves the most numerous, successful, and diverse groups of animals on the planet. which means that this random bacterium that this duo found is arguably one of the most successful microbes on Earth. And what it does varies depending on which animal it's found in. So in some insects, Wolbachia has a problem with males because it only passes down the female line from mothers to daughters. And so sons and males are a dead end to it, and it has many ways of dealing with them. In some insects, it transforms the males into females as they develop. In others, it just outrightly kills male embryos before they even hatch. In some insects, it allows females to reproduce asexually, cloning themselves, so they have no need for males whatsoever. It's the ultimate warrior against the patriarchy. In other animals, Wolbachia is more of an ally. It provides bed bugs with vitamins that are missing from their meals. And in one of my favorite examples, it allows the caterpillars of one particular moth that feed in the leaves of trees to stop those leaves from turning red during the fall. It creates these little green islands in which the caterpillars can continue feeding in peace while the world turns red and yellow and orange around it. But the insect that matters most to us for our story is one called Aedes aegypti. It is a mosquito that spreads diseases such as Zika, dengue fever, yellow fever, chikungunya, and many others. Aedes aegypti does not naturally carry Wolbachia. It is one of the few insects that does not. But for the last several decades, Australian scientists have been trying to get the two of them together, to get to the mosquitoes, to stably house Wolbachia inside their bodies. Why? Because if Aedes carries Wolbachia, it cannot spread the viruses behind all of those diseases that I talked about. Rather than becoming a vector for things like Zika and dengue, it becomes a dead end. And because Wolbachia is so good at influencing the biology of its hosts, it has a tendency of spreading very, very quickly. So if you release a small number of these Wolbachia-infected mosquitoes into the wild, very soon the entire wild population of mosquitoes will be infused with these microbes and therefore be unable to spread human diseases. They don't die, no microbes are harmed in the making of this technique, but they stop becoming problems to us. They'll bite, but they won't spread disease. And this approach has been tested in Australia. It is now being trialed on a much larger scale in places like Vietnam and Indonesia and Colombia and Brazil, and probably soon in parts of the US as well. And it has tremendous potential for controlling diseases like dengue and Zika in a way that insecticides do not. Insecticides are expensive things that must be continuously resprayed in order to be effective. They are also toxic and they cause side effects. Wolbachia has no side effects. It is not toxic. Once you release the mosquitoes, you're off to the races. You don't need to keep on doing it. So it is extremely cost effective. Who's to say whether this technique will work at a large scale, but I think that it has a lot of potential. And critically, none of this could have been anticipated back in the 1920s when Hertig and Walbach made their discovery. In fact, Walbach died in the 50s before anyone realized how common Wolbachia was, before he never had a chance to appreciate that his name had been attached to this thing, which was one of the most successful microbes in the history of the planet, let alone one with the potential to influence and help hundreds of millions of people. And that, I think, is the story of the microbiome in a nutshell. For the longest time, we have neglected and feared microbes, and only recently have we come to appreciate them for the important part of the world around us that they surely are. And only recently have we gained enough confidence to try and manipulate these worlds for our own gain. Now, as I said, our attempts at doing so are still very basic and very stumbling, but there is a huge amount of potential there. And I think we will only tap into it if we understand more about this world around us, and if we feed that curiosity to learn more about the planet, about the things that we share it with, and about the things that we share ourselves with. And that is why I wrote I Contain Multitudes. Thank you very much. And thanks. So we have time for some questions. Mike there, Mike there. Hello. Can we get... You go and then I'll repeat it.
About communication. Yeah. Apparently, microbes in our gut communicate with one another. Can we hope to communicate with them?
Okay, the question is whether we can communicate with the microbes in our gut as they surely communicate with each other. The answer is that we do all the time. Without knowing it, of course, we exchange signals with the microbiome within us. That is how we regulate their communities and they influence us. You know, the interactions I talked about with the immune system, our immune system is constantly influencing and changing the species and strains of microbes that live within us. The genes that they switch on, the molecules that they produce, they in turn, through their relative abundance and through the chemicals that they produce, influence the way our cells react. So there is very much this ongoing negotiation. We are not privy to it. It happens below the level of our awareness, but it definitely goes on. And we can certainly hope in the future to tap into those communications and to steer it in a more directed way by understanding the way it already works within us.
Thanks.
Hi.
My name is Alan Steer. Thank you very much for a very lucid and clear presentation of a complex subject. The point I'd like to ask about is having you enlarge upon actually what you were just talking about, and that is the influence of our own immune system on the makeup of the microbiome, and I'll particularly refer to the gut microbiome. I'm asking this as it relates to disease, such as inflammatory bowel disease, rheumatoid arthritis, where dysbiosis develops. And you mentioned the animal models, and there are some animal models of both of those diseases, albeit imperfect. When one transfers microbiome from one to another, it doesn't cause disease by itself. Something has to be wrong with the epithelium. So in the animal models, one is generally doing something to damage the epithelium. Of course, in humans, that's presumably not the case, and it's our own immune system, probably, that's having some role in changing dysbiosis.
It's a lot of different factors, and you're right that it is unlikely to be just a change in the microbes that causes problems. It is likely to be a number of different hits. So maybe there have been some great studies showing examples where you have, again, animal models where you have a genetic susceptibility, a prior infection, a change in the microbe, a dietary change, something like that. So the big questions, I think, are what does it take to set a microbial community off course? You know, is a strong dose of antibiotics enough? Probably not. Is a change in our diet enough? Is a genetic predisposition to a disease enough? It's probably a likely combination of all of these things. And of course, these things are cyclical. So our bodies and our lifestyle influence our microbiome, and they influence us in return. So with the immune system, one of the ideas that has risen to prominence and has a lot of evidence behind it is the idea that without exposure exposure to a wide variety of microbes at an early age, we fail to educate our immune systems properly and teach it about the wide variety of organisms that usually would share our bodies. And so when we grow up, we develop hyperactive, twitchy immune systems that overreact to things in the world around us, whether it's stuff, organisms that live with us, whether it's dust or pollen and so on, which might explain part of the rise of things like allergies and asthma, inflammatory diseases, autoimmune diseases. So these are very complicated things. They're cyclical things. They involve a lot of different factors acting in concert. And I think teasing them apart and also looking at them together is the big challenge for microbiome science going forward.
Thank you.
Hi.
Hi, thank you very much for your talk. I'd like to get into more specifics. In the healthcare system of the United States, you can basically look at the cost at four or five major problems: diabetes, obesity, heart disease. We can choose those. But obesity, which can lead to an awful lot of these other diseases, is one of the end results that's being attempted to be treated by the biome of the body by various companies. Can you tell us where we are in the development and commercialization of the products dealing with the issues such as obesity, ulcerative colitis, and diabetes?
Yes, we are at a very, very, very early stage. If someone is telling you that they can change your microbiome in order to make you thinner, they are lying to you. I'm going to give you more detail because you asked for it. The obesity is the condition that has been most heavily studied with relation to the human microbiome. There were seminal studies published in 2006 showing that obese and lean individuals, humans and rodents, have changed differences in their microbiomes, and that if you transplant microbes from a fat mouse to a lean mouse, the lean mouse will put on weight, which show people, there's causal effect here, maybe by doing similar transplants the other way or by influencing the microbiome in some way, we can make people thinner in a way that will be much easier than having to do all the boring things like eat healthily and exercise and so on. There are a few problems with that. Firstly, as more and more studies have turned up, a lot of those early results haven't replicated. They haven't checked out over time. So one of the classic examples is it turned out early on that it looked like obese individuals. So in the human gut and in the gut of many other animals like rodents, the two major groups of bacteria are the Bacteroidetes and the Firmicutes. Just call them B and F. And one of those early results said that obese individuals have more B to F. No, the other way around, more F to B. This was a classic result in the field, and it doesn't shake out over time. You look at studies now, and you don't see that F to B ratio holding up when you look at larger samples of people. And indeed, in those early studies, you can find differences in the microbiomes of lean and obese individuals, but those are not the same differences that you see across the obese and lean individuals from different studies. So you can always find differences in the microbiomes of people depending on their weight, but it's very hard to show consistent differences, which means, for a start, the microbiome is not a predictive science yet. I cannot look at your microbiome and say, you are going to be obese and you will not be. That sort of thing has been worked quite well in some studies, but won't work like just in the general population. And nor is it a thing that we can tweak and manipulate. We don't know how to do that well or effectively yet. And we're still at that early stage, despite the fact there has been, now coming up to over a decade to work on it. So obviously there is a huge amount of interest here. But the other point is that you can't just make people thinner by changing their microbes. Even in the animal studies, animals that receive the microbiomes of a leaner individual only lose weight if they also eat a healthy diet. So bad news, everyone, there is no shortcut to being thin, but there is this microbial component that is relevant. We just don't know how to tweak it.
Thank you.
Thanks. Hi.
Hello, can you please tell us more about viruses, like the relationship between viruses and bacteria? And
Yes.
if there are helpful viruses?
I would be delighted to. So viruses have an even worse rep than bacteria do, but the vast majority of viruses do not infect us. They actually infect bacteria. Most of them are called bacteriophages, or phages for short, and they also live within us. I said we have trillions of bacteria inside our bodies. We have quadrillions of viruses, phages, and others. And they are not making us sick. They are living within us. Some people believe that some of these phages are part of our immune system, that they actually contribute benefits to us, that they may be part of the ways in which we control the population of microbes inside our gut, or that one of the ways in which we select which species and strains get to live within us. There are also other examples in the animal kingdom where animals have harnessed viruses for extraordinary purposes. So the animal that was on my podium was a parasitic wasp. There are some wasps that have incorporated the genomes of viruses into their own DNA, and that now build domesticated viruses with which they use to attack the insects that they parasitize. So they've turned viruses into essentially biological weapons that they build themselves.
Wow.
All this and more in the book.
Next book, thank you.
Hi.
I like your shirt.
Thank you.
I have two questions that are kind of related. I read that the appendix is actually there to repopulate the gut in people who get sick with something like C. diff, and that people who get C. diff, if they've had an appendectomy, they don't heal as easily. And so then, too, I wondered if it would be a good idea for when people are young and vital and healthy, that maybe they should bank some of their fecal material for in the future if they...
So the appendix idea is certainly something that I've heard before. It's quite an old idea. It doesn't have the hugest amount of evidence behind it. I think there is some, but it's certainly not a theory that the microbiome research community seems to be taking hugely seriously. That doesn't mean that it's wrong, and I only know a little bit about it, so I don't want to overly downplay it to you. But it's it is what it is. Maybe some people will look at it in a bit more detail in the future. Whether we should bank our own young poo. There are actually stool banks out there. There are companies, some non-profit, some for-profit, that provide frozen fecal samples for the purposes of doing fecal transplants. And there are very good reasons for this. This is a very simple procedure to do, but it is not a procedure that should be done lightly. Through a fecal transplant, you can transmit all of the diseases that transmit through blood products, like HIV, hepatitis. We have seen, as we have discussed before, that you can transmit the symptoms of certain conditions through moving microbiomes from one individual to another. And so when these procedures are done in hospital settings, they are done under very, very strict screening procedures for the donors. People have to be very healthy. I think a lot of places now won't allow you to donate stool if you are obese, if you have inflammatory bowel disease, if you have any of these conditions. So you're looking for like super healthy people. So some places are thinking, okay, let's collect a lot of their poop and bank them for future use. Thanks.
Just make one suggestion. Just making one suggestion, because these are fascinating questions. Let's take all the questions that are for people who are currently standing, and those of you who are waiting to get books signed, will be maybe 5 or 7 minutes getting over to the Alumni Hall to sign books. Is that okay? Yep. Thanks.
Let's go. I'm going to write down your questions. Hi.
Hello. So this is kind of a what's the current state of the science question in connection with sort of gut science and mental health.
Okay.
We often talk. You touched on this very briefly, but we often talk about something like depression as, a lack of or a chemical that isn't, a chemical imbalance. I know that there's more science related to this happening now. Like, what's the theory versus the fact on the relationship between mental health and?
Got it? Yep.
Yep.
Chemical imbalance, mental health. Okay. Hi.
Hi.
I'll take all of these and then we'll do them all in one fell swoop.
Okay.
We're going to take all the remaining questions and then I'm going to answer them again. So,
Okay, well, you
shoot.
mentioned that we all have different microbial patterns within us, profiles within us, and that, and you've also talked about genetic differences. We all know that certain populations are more prone to some diseases than others. Have they found differences in the profiles of the generic profiles of microbes within different racial, geographical, ethnic groups, and could that be harnessed to prevent diseases?
Okay, yep. Thank you.
This ties into what Amanda said earlier. There have been reports that the sweeteners in diet soft drinks actually alter the gut microflora, leading to such things as diabetes and obesity. From what I've read on the web, some people say yes, some people say that's unconfirmed. What's the current thinking on that topic? That's simply my question.
Got it. Okay. And last question.
In terms of evolution, would Lynn Margulies' theory of symbiotic genesis be closer to how we might have evolved with the microbiome versus Darwin's?
Okay. Yep. Great. Okay. That's a good question, you guys. All right, mental health. When we talk about microbes influencing brain and behavior, it does seem a little bit weird, like how does what happens in the gut influence the brain? Actually, there are a lot of connections between the two. So there is the vagus nerve, which runs the full length between those two organs and acts as a kind of phone line between them. The microbes in our gut actually produce a huge amount of what are traditionally called neurotransmitters, so things like dopamine and serotonin. They are not just chemicals that circulate in the brain and influence behavior. They also are made in substantial volumes in the gut as well. And the immune system acts as a sort of conduit between the two. So there are many ways in which the microbes in the gut can influence the brain. And a lot of animal studies showing that there is an effect. Now, there are inconsistencies there, as there always are in the early stages of any field. So again, it is difficult to say to what extent this applies to us, to what extent it influences our mental health, or to what extent it can be altered to improve our mental health. I'm very comfortable saying that the microbes within us can influence the way we behave and the way we think and the way we feel, but the absolute specifics, like which strains does what in which people, what can we do to make someone feel better? I think these are, again, unanswered questions, but an area of very active research. Okay, differences between geographical populations. Yes, we have to be very careful about this because often if you compare people from very different parts of the world, it's very easy to commit a fallacy in thinking that like the differences between trait X might explain why one population is more likely to get a disease than another, whereas there are so many differences between people from different backgrounds and from different geographical regions. That being said, when we look outside the Western world at a lot of hunter-gatherer populations from all around the world, from Papua New Guinea and Colombia and Kenya and so on. What we see consistently is that there is a greater diversity of microbes in those people than in us in Western urban settings. So a wider range of species. What is unclear is whether that is a good thing or a bad thing or just a thing. Because we know that our diets influence our microbes to a huge degree, and people who are hunter-gatherers eat a very different diet than what we eat. They eat a wider range of plants, for example. You eat a wider range of plants, you will have a wider range of microbes. So whether, the microbes that they carry aren't necessarily all going to be good. Some of them might cause diseases, they might have intestinal parasites, all of this sort of thing. Some people have put forward the idea of doing fecal transplants with hunter-gatherer stool. I think this has a really uncomfortable connotation of colonialism and the noble savage stereotype. But it also probably just won't work because these things vary so much from person to person. It's all very circumstantial and contextual, and we still don't understand those connections enough yet. Soft drinks. There have been studies suggesting that both soft drinks and a lot of modern products like emulsifiers in food or artificial sweeteners can change the composition of the microbiome, can lead to what is often called dysbiosis, and that might increase the risk of things like diabetes and obesity and so on. I think a lot of those studies have been done in animals, so they need to be confirmed in humans. They are, you probably sense a recurring theme of what I'm going to say, preliminary work that is very interesting and that we need to check out in larger studies and in people to work out how relevant it is. I think that you can do large epidemiological studies finding links between consumption of soft drinks or processed sugars or whatever and risk of obesity and diabetes and so on. The microbiome may explain part of that, but it's not an explanation in its own right. If you are saying that the microbiome explains the connection between thing X and disease Y, you then also need to explain why thing X changes the microbiome and then why the microbiome influences disease Y. It's not an explanation in itself, it demands more explanations in its own right, and that's what we need to get more of. And finally, Lynn Margulis and her ideas of symbiogenesis. So for those of you who don't know, Lynn Margulis was an iconoclastic late scientist who did not come up with, but certainly championed the idea that a lot of important evolutionary events came when microbes fused together. And she is partly right. She's particularly right in one very important way. All of our cells contain little bean-shaped structures called mitochondria, which provide us with energy. They are little power plants without which we would be unable to survive. Mitochondria are found in all of our cells, in every animal, every plant, and so on. Mitochondria were bacteria. They are the domesticated remains of bacteria that were gobbled up by another ancient microbe and became a permanent part of it. So aside from all the microbes that we carry in our mouths and guts and skin and so on, every single one of our cells has domesticated bacteria inside it without which we could not survive. And Margulis made a very strong case for the fact that mitochondria were in fact formed bacteria, and she was vindicated after much opprobrium and disbelief. But it is a common misconception that this idea of symbiogenesis it contradicts Darwin's idea of evolution. It in fact does not. It just adds another twist to it. In fact, the microbiome doesn't really alter anything about the way we think evolution behaves. It means that it gives us an understanding that evolution may occur sometimes much faster than we think it does, but it still operates in the same Darwinian way. Like I told you about the insects that can become instantly resistant to insecticides if they swallow the right microbes. That sounds a bit anti-Darwinian because it's instant, it's very quick. But remember that the adaptations that allowed the bacteria to detoxify those insecticides happened in the usual Darwinian way. Slow build-up of changes over time. It's just that the animals get to benefit from those changes instantly by forming partnerships with those microbes. The last thing I want to say about this, which is kind of cool, the idea of mitochondria being formerly bacteria may actually be really, really important. It may be one of the most important moments in the history of life on Earth, because for the first billion years of life, things were just bacteria or other single-celled tiny microbes. And all of this that you can see, all of the animals and plants, algae, fungi, and so on, these are organisms called eukaryotes. We only appeared much, much later, and it took a long time for us to show up. Some people think that's because our cells are much bigger than those of bacteria, and our genomes are much more complicated. And we could never have achieved that complexity without the energy that mitochondria provide to us. So it was only through one fluke moment when one microbe ended up inside another microbe, forming what would be the ancestor of all of our cells and of the mitochondria within them, that allowed complex life to occur and to evolve and to thrive. It was a freak moment, this weird partnership that some people think happened only once in 4.5 billion years, and without which none of this would be here, which is just something to think about when you're going around looking at nature. All right, thank you very much.